So good morning. Today is Friday, June 25th. We've made it through another week, or almost made it through another week. I guess we haven't technically made it through it yet, but we have gotten to the end point. We can see the finish line from where we're at. And we're going to basically continue what we were doing in the last class. We're going to spend lecture talking about physiology and our lab talking about the bones and bone features. I again want to thank the groups for going first. Again, not that they had a choice on it, but I appreciated what they did. And again, I think everybody did an excellent job. But the one thing that I would point out is we did run a little long. And I think one of the reasons for that is that I really want people to focus just on the bones and bone features that are on your list. While everybody provided great information, it can be sometimes a little overwhelming when you're providing all that information and it isn't stuff that's going to be on the exam. With us having such a streamlined summer class, I think it's really important to focus just on the content that's going to be on the exam. So I would encourage people to really focus just on the stuff that you're going to be held responsible for on the exam, and I think hopefully that will help. And so we'll do the rest of our skeletal. We need to finish off the rest of our skull features. So we will start that. And then from there, we will go into the axial skeleton and finish the rest of that today. The groups that are going to be presenting the vertebral column and the bony thorax. Also, I will assign your appendicular bones and bone features that you're going to be responsible for next week. And we'll be doing those on Monday and Tuesday. Also on Monday and Tuesday, your Unit 8 review is going to be due and your Unit 9 review is going to be due. And then also on Tuesday, your 30-point skeletal review is due. And remember, that will be graded for correctness. All to help us to prepare for our Thursday exam. We have our lab and lecture exam on Thursday, the 1st. And even though it's the 4th of July weekend, we don't get a huge holiday. So Friday, we do have to turn around, come back, and we are going to start the muscular system. It is an important lecture. This lecture on Friday is where we're going to do most of the anatomy of the both gross and microscopic anatomy that we are going to be using for what I believe is the longest physiological process we will be talking about in this class. In fact, it is so long, we are going to be taking the process by which a muscle contracts, and we are going to divide it up into 3 slightly more easily digestible pieces. And I guarantee you that there will be at least 2 essay questions on those 3 processes on the exam. So it is very important to be here on Friday so that you can get the anatomy so that you can understand the physiology when we come back. Now with the 4th of July, we do get a holiday. Our holiday is not the Friday, it is the Monday. So Monday the 5th, you get to sleep in after all of your drunken debauchery on the 4th. And then we will meet back on Tuesday the 6th. All right, that is the game plan. Questions on any of that? All right, let's go ahead and pick up where we left off with the lecture. I know last time I said we would finish with the, we would start with the skull features, but I think it still makes more sense to save that for the lab. So when we'll start the lab with those, I'll finish up the rest of the presentation of that, and then we'll talk about the rest of the axial skeleton and go on from there. So if there's no questions, we will dive back into lecture. All right, excellent. So we left off last time. We were working through our way through all of the physiology associated with the bone. And last time we had left off with our normal bone development. There were 2 methods of making bones. And what were the 2 methods of making bones? Give me one of them.
Endochondral ossification.
Excellent. And what's the other one?
Intramembranous ossification.
Excellent, excellent, excellent. So there were 2 possible ways to make bones. And of course, as we talked about, that is going to lend itself to 3 potential essay questions. We also mentioned that there are 2 normal ways to grow the bones. And anybody remember the 2 normal directions we need to grow a bone?
Length and width.
Length and width, exactly. And so that's where we're going from here. So let's start with that. Starting first with bone growth in length. Where does this bone growth in length take place?
Epiphyseal plate.
There we go. We know in that hyaline cartilage, that is the epiphyseal plate, is where our bone is going to grow in length. And we also actually know how as well, right? That epiphyseal plate is made of hyaline cartilage, and we know hyaline cartilage, like many tissues, can grow two ways, from within the tissue or by putting more layers on top. And which way did we say we were going to use for our bone growth in length?
Putting the layers on top.
It wasn't the layers on top, it was growing it from within. And which growth process was that?
Intramembranous.
Close. It is interstitial. Remember, interstitial growth is when the chondrocytes divide inside of the tissue, make matrix, and push away from each other. All right. So this bone growth in length is going to take place in the epiphyseal plate, and it's going to use the interstitial growth. So let's go to the whiteboard and start there first. We'll do this really, really simply. Here is the diaphysis of the bone, and here is the epiphysis. And of course, I'm going to tremendously exaggerate the epiphyseal plate in between them, just to make my point. So here, this open space is our epiphyseal plate. Again, this is an incredible exaggeration, but it allows us to expand it and be able to see, and certainly isn't drawn to scale. But let's talk about what we're doing here. Growth in length. And again, it uses interstitial growth. It occurs in the epiphyseal plate, which we know is hyaline cartilage. Now, this growth is seen in actually 5 distinct layers. I know we think of this as a tiny little line, and indeed it is a tiny little line, but there are actually 5 distinct layers or zones to this epiphyseal plate where the growth is taking place. The first of those happens to be a region that is known as the resting zone. How much active growth do you think is taking place in the resting zone?
0.
Exactly. The resting zone isn't active in the growth process. But it is living hyaline cartilage. And so what do you think its function is then? Well, it turns out basically it's just the anchor for the epiphysis. Basically just helps to hold the head to the rest of the growth plate as it is taking place. After all, we don't want the head of our bone wobbling around on top of the diaphysis. We want to make sure that it is anchored in place. So we have our, all right, put a couple lacunas in here, put a couple chondrocytes in here, and we just have this resting hyaline cartilage in this zone. Excellent. There you go. Not too terribly exciting. But let's talk about what's happening in the next region. The next region, and I'm going to cheat and I will just use a box here. Excellent. Oh, sorry, hold on. Is what is known as the proliferating zone. Guess what's happening in the proliferating zone?
Actively dividing.
Exactly, and that's exactly what's happening in here. Here is where we have our actively dividing chondrocytes. Remember, as we talked about, the chondrocytes are actively dividing, they're making new matrix, and they're pushing away from each other. This is happening so rapidly that essentially what starts to happen, and I'm not going to bother drawing the chondrocytes inside of it because it'll simply take too much time, is that we start to get these columns of chondrocytes that start to appear. As they start to push and divide, they kind of look like stacks of coins where they're constantly dividing, constantly pushing away from each other. And it is actually here in the proliferating zone that our tissue is actually expanding. Right? If you think about it, because these are pushing away from each other, this is where we get the expansion of the tissue. And again, using that interstitial growth that we talked about. And the epiphysis is moving farther away from the diaphysis. Excellent. Mission accomplished. Just like that, we're done. Right? Because now our bone's getting longer and that's all we care about?
No.
No, why not? The bone's getting longer. That's our goal.
There's 3 more layers.
True, there are 3 more layers. So obviously that's an indication that something is going on. But let's also think about this. In your mature adult bone, is your mature adult bone just a small little basic chunk of bone? And then on top of it at both ends, we have huge chunks of hyaline cartilage. And then the epiphysis at the end of those. Is that what a mature bone looks like? Whoops, hold on.
No.
No. So while we are indeed making the bone longer, we're making the bone longer with hyaline cartilage. Right? So obviously we're not fully done yet, but the good news is, do we know how to convert cartilage into bone?
Yeah.
Absolutely. That is a process that we have definitely seen. So let's think about that. What's the first thing that has to happen if I'm going to convert cartilage into bone?
Blood vessels got to come in.
Well, but if the blood vessels are going to come in, we need there to be a space for those blood vessels. And how do we make that space?
With some osteons.
True, but there's all these pesky chondrocytes in this place. We have tissue now that is filled with chondrocytes, right?
Do the chondrocytes have to die?
Excellent. And what do they do before they die?
They grow
Enlarge.
and
Bingo. There we go. Excellent. The next zone is what we call the hypertrophic zone. Right? Hyper means what?
Excessive.
Excessive or increased. Absolutely. Anyone know what trophic means? Trophic refers to growth. And so that's exactly what happens in this area. In this area, our chondrocytes enlarge. And as we've learned, when our chondrocytes enlarge, obviously their lacunas have to enlarge to make space for them. So we start to get these really big chondrocytes in this region. And the problem with that is as we get these really big chondrocytes, they take up more and more of the matrix, and our matrix thins and hardens. So here in this region, our chondrocytes enlarge in size. And as they enlarge in size, the matrix thins and hardens. And that becomes an issue because as that matrix thins and hardens, what ends up happening to our chondrocytes?
They die.
Our chondrocytes die, exactly. Chondrocytes die. So let's do that. Matrix calcifies or hardens. Chondrocytes die, leaving a cavity. And then, as you mentioned, once we have a cavity, blood vessels can grow in. And when those blood vessels grow in, they bring in new cells, they bring in new chemical signals. This region where the cells die and the blood vessels grow in is what is known, oops, that should be there, as the calcification zone. And then, of course, once those blood vessels grow into this area, let's go ahead and draw some calcified looking matrix and bring some bright blood vessels into the area. They bring the chemical signals. They bring that's too close to the red. Always pink. They bring the osteoblasts, and then we get our ossification zone. And in the ossification zone, this is where the osteoblasts and osteoclasts remove the calcified matrix and replace it with bone. And so in that fashion, we have both expand the tissue with cartilage and then converted that cartilage into bone. Now, again, of course, I've done an amazing job of drawing this on the board, but let's look at the pretty pictures from our textbook. Again, our goal here is to grow the bone in length. This happens in the epiphyseal plates for our long bones. As we know, that plate closes sometime between 18 and 24 years of age, at which point our bones have reached their ultimate length. They cannot get any longer after that. And that epiphyseal plate has, as we mentioned, 5 zones. So I've done a horrible job of drawing it on the board, but let's actually look at what this looks like under a microscope. Under a microscope, if we look at that small line of hyaline cartilage, we can see indeed that there are 5 distinct regions. Up here, just below the epiphysis, is that resting zone where we just have normal looking hyaline cartilage. Again, this does not play any role in the growth process, but it does play an important role in that it helps to anchor and hold the epiphysis in place. The growth in length occurs in this second region. Right, it's numbered 1 because, again, it's not playing, the resting zone is not playing a role in the growth process. I don't care how you label them, 1 or 5, 4 or 5, but the point is here, notice we see the rapidly dividing chondrocytes, and they are stacking up like coins because they are rapidly dividing and pushing away, and then dividing and pushing away, and dividing and pushing away. The key is this here is the area where the tissue is expanding, where the tissue is getting bigger. So right here in the proliferating zone is where the epiphysis moves further away from the diaphysis. The problem with that is while the bone is getting longer, it is getting longer with cartilage. And so we need to then convert that cartilage into bone. And we know that's a 3-step process. It starts first with the chondrocytes getting larger. Notice how much bigger these chondrocytes are getting. And as they get bigger, the matrix in between them thins and it hardens. And as it thins and hardens, it gets harder to get nutrients and oxygen to those cells. It makes it harder for those cells to get rid of their wastes. And so ultimately, they die. When they die, they just leave that hardened, calcified matrix, a big cavity in there that our blood vessels can grow into. And when the blood vessels grow into here, they can bring osteoblasts and osteoclasts. The osteoclasts break down the calcified matrix, and the osteoblasts replace it with bone matrix. And so if you think about it, here is my epiphyseal plate. Using interstitial growth in the proliferating zone, I make the plate wider, and then I convert some of that plate into bone. And then I make it wider, and I make bone, and I make it wider, and I make bone, and I make it wider, and I make bone. Notice in this fashion, the size of my epiphyseal plate really doesn't change because I'm making more cartilage, then converting cartilage to bone, making more cartilage, converting it to bone, but my bone keeps getting longer. All right, questions on this?
Yeah, I had a question.
Yeah.
So how exactly does the calcium get hardened, the matrix? Is it the cells that are pushing against each other, creating a lot of pressure? And does that kind of harden?
Yeah, part of it is as they expand, you're right, they're putting pressure and condensing the matrix. And basically, as they condense the matrix, they're pushing water out of it. And so as they push the water out of it, it kind of dries and it kind of hardens. And as it dries and hardens, it makes harder to get nutrients in and out, to get waste in and out of that matrix. Exactly. Great question. Any others?
Yeah, you were saying that when the blood vessels come in, it brings osteoblasts, it turns osteocytes. Now, does it bring it in, or is there mesenchymal cells that just get turned?
Great. The blood vessels have to bring it in, because remember, in this case, when that little region of the epiphyseal plate calcifies, as a result, the cells inside there die. So there aren't, not only are there no chondrocytes, there wouldn't be any mesenchymal cells in there as well. So basically what happens is we bring new mesenchymal cells in with the blood vessels, we bring the oxygen and nutrients for those, and then we bring the chemical signals to tell those mesenchymal cells, all right, I need you to make osteoblasts and I need you to make osteoclasts so that we can break down this calcified matrix and we can replace it with bone. So yeah, the blood vessels growing in have to come in to bring those new cells into there. These are great questions, but you guys haven't asked the one that I thought you would ask. Remember,
What tells it to stop?
Exactly, right? If we are making more cartilage and then converting it to bone, and making more cartilage and converting it to bone, and making more cartilage and converting it to bone, and because of that, the epiphyseal plate never changes size, why do we stop growing in height? What makes this epiphyseal plate close? So I'll ask you that. What is it that you, go ahead.
I was just say a chemical signal at the proliferating zone.
Well, you are close. It is indeed a chemical signal. What chemical signal or signals do you think it might be? What changes our rate of growth? What biological process that all of us have gone through changed your rate of growth?
Puberty.
Puberty, absolutely. And during puberty, we produce an increase in our sex hormones. What are the two main classes of sex hormones?
Estrogen and testosterone.
Well, careful. Estrogens is a class of hormones, almost a dozen hormones, the most common of which is estradiol. And androgens are the class of hormones in males, but you're absolutely correct. The most common androgen is testosterone. So we have these two sex hormones, estrogens and androgens, and it turns out what they do is they stimulate osteoblasts. And they make osteoblasts make bone faster. So we hit puberty and we get this growth spurt. But while we're getting our growth spurt, something else is happening in our epiphyseal plate. In our proliferating zone, we're making more cartilage, and now these stimulated osteoblasts make more bone. And then we make more cartilage, and we make more bone, and we make more cartilage, and we make more bone. Notice now my epiphyseal plate is getting smaller and smaller until finally those superactive osteoblasts ossify all of the cartilage. And when they ossify all of the cartilage, suddenly our epiphyseal plate is gone, and now we have an epiphyseal line. And I have one more question for you. Do both estrogens and androgens stimulate osteoblasts equally?
No.
Right, I'm asking the question, so clearly the answer is no. Which one stimulates the osteoblasts more?
Estrogen.
Why do you say estrogen?
Process of elimination. Women tend to be shorter.
Bingo, exactly. You are absolutely correct. Estrogens stimulate the osteoblasts more. And you're absolutely correct. There are two huge implications to that. The first, as you mentioned, because it stimulates them more, women tend to be shorter than males. Now, again, height is one of those things where there's huge variance in, right? Even in a class this small, is every single male taller than every single female in a class like this? No, of course not, right? And so there's huge ranges, but in general, you are right. Estrogens stimulate osteoblasts more, so their growth plates tend to close earlier, and they tend to be relatively shorter on average than the average male. But there's another huge implication as well. Notice both males and females increase estrogen and androgen levels when they reach puberty. And then the levels of both stay high forever until 15 minutes after they're dead?
No.
No, in males, they do. Males basically produce androgens, and androgen levels pretty much stay high. They may drop a teeny bit, but they pretty much stay high until 15 minutes after they're dead, right? But what about females?
Drops off.
Right. Females
Significantly.
enter puberty, estrogen levels stay high, and then what happens?
Menopause.
Menopause. And what happens in menopause?
Stop producing.
Right. There's a huge drop in estrogen levels, right? Again, one implication for that is basically females become infertile at that point, right? From a biological standpoint, they're now unimportant, right? And again, just from a biological standpoint, can't reproduce, right? But more important than that, remember these estrogens are stimulating osteoblasts to help not just to grow, but also to maintain bone density. So if suddenly those estrogen levels drop, all that estrogen that was stimulating the osteoblast to maintain bone density, what is much, right? Women are much more likely to lose bone density after menopause as they age than males are, right? What do we actually call that condition?
Osteoporosis.
Osteoporosis, absolutely. Females, right? Grandma can be drinking a whole cow's worth of milk, but if she doesn't have those estrogens to tell her osteoblast to take that calcium and put it into her bones, grandma could still be losing bone density as a result of that. So absolutely, osteoporosis is a much more serious concern in females than it is in males because of these fluctuations in hormone levels. Excellent, excellent. Questions on that? All right, spectacular. That is how we grow a bone in length. So let's switch and talk about how we grow a bone in width. Let's cheat. come back here. I'll go ahead and save this even though the picture's silly. And let's talk about our growth in width. The growth and width of our bones is going to use what growth process?
Intermembranous.
I'm sorry?
Interstitial.
Well, remember,
Ossification.
interstitial, we grew the tissue from within, and that's how we grew in length. So what was the other growth process?
Intermembranous.
Well, again, be careful. Intramembranous and endochondrial is how we make bones. Those aren't the growth processes we talked about. The growth processes that we talked about, there we go, were interstitial and appositional. Appositional, remember, is where we add new layers on the outer surface. Now, if we're going to add new layers of bone, because these are bones that are already here, if we're going to do this in bones, this has to occur on the outer layer where the immature cells are. So that's going to be under the periosteum. However, if you remember, we also said this appositional growth on the outer surface of our bones is not only how we make the bones wider, but it's also how we form our osteons, right? This is how we form our compact bone. All right, let's start with a very simple picture. Here we have a cross section through a bone. All right, it's a long bone, so we'll give it a little bit of a medullary cavity. And we know that there is, actually, let's just cheat and do it this way, a periosteum on its outer surface. All right, this is basically in its simplest form what a long bone looks like if we cut it in a cross section through the diaphysis. Now, if we cheat and expand out my periosteum, get it out of the way, we know that under this periosteum are going to be the mesenchymal cells. And we know mesenchymal cells divide to produce osteogenic cells that are the unipotent stem cells that divide to become osteoblasts. So that mesenchymal cell becomes an osteogenic cell, becomes an osteoblast. And then we know this osteoblast builds matrix and surrounds itself with matrix. And once it surrounds itself with matrix, it matures into what?
Osteoclasts. They're osteocytes.
Yeah, into an osteocyte. Excellent. And essentially, this process, and I'm going to totally cheat and get rid of my, get rid of my periosteum entirely. Because then what that will allow me to do is expand that layer all the way around. And notice as I continue to make those osteoblasts that make matrix and completely surround it, basically I form a layer of matrix around the entire circumference of the bone. So in this process, I form a continuous layer of matrix around the entire circumference of the bone. And what would that be called?
Circumferential lamella.
A circumferential lamella, absolutely, right? And then since it's still under the periosteum, can it happen again? And then again, and then again? Absolutely. And notice what's happening as I do this. As I make these again and again and again, my bone gets wider and wider and wider. Once again, super simple process. Except that's really not what it looks like. Because as we saw, when you cut through a long bone, does it look like the ring of trees where we're able to just count the number of rings and then we can tell how old you are? Is that what our long bones look like?
No, just the outer layer.
Right. There are a few of these on the outer layer, but this is not how our bone is organized. So the good news is this is exactly how we're going to make the bone grow in width, right? Basically, growth in width, we are adding new matrix to the outside of the bone. And as we do that, the bone gets wider. Right? So at its core, this is it. It is really just that simple. However, as we know, nothing in life is really that simple. And so there's more to it. There's got to be a kink in the system. So in this case, what is the kink in the system? What's lacking from this picture? Well, remember, one of the things that we have a ton of associated with our bones is blood vessels. So it is the blood vessels that disrupt this system. And while they disrupt the simple system, the advantage of that is it actually is then going to cause the formation of our osteons. So let's take a closer look at this process and see how it works. Now, to do that, what we are going to do is rather than looking at the entire circumference of the bone, I'm just going to take a wedge of it. If I just take a wedge of it, this will take us a closer up view and give us a better idea of what's going on. So let's go ahead and erase all of this. Actually, those I should probably have left. I'll sneak this down here. Sneak that down there. Remind us of that there. And over here in the corner, I'll draw that wedge of my bone. So here is that wedge of my bone. Here is our medullary cavity. Which of course is lined with an endosteum. So let's go ahead and draw that in purple. And again, we know it's in direct contact, but I'm leaving a little bit of space here. And we also know here on the outside, we have a periosteum. Now, for simplicity's sake, I'm going to not draw the periosteum the rest of the way around. But we do know that the periosteum is there. And we do know the periosteum houses our mesenchymal cells. So I'll draw that part here. And then that way I don't have to worry about drawing it here and moving it and erasing it and doing all of those fun things. So we have our periosteum with our mesenchymal cells. All right. But what we hadn't drawn before, and we will draw now, is an example of a blood vessel. Here is a blood vessel from the periosteum. So remember, in our periosteum, we also have blood vessels. And that'll be an example of one. Now, as we just finished talking about, we know that that mesenchymal cell is going to divide to become an osteogenic cell. That osteogenic cell is going to divide to form an osteoblast, and that osteoblast is going to surround itself with matrix. And so as it does that, as we've talked about, it will form that circumferential lamellae on the outer surface of the bone. But what's interesting is as these circumferential lamellae form, they have to respect the boundaries of the blood vessel. So they don't grow where the blood vessel is. So as I continue to make new layers, increasing the width of my bone, I start to form what we call these periosteal ridges. So what ends up happening is as we're making this new matrix, as we're adding new layers on the outside, what normally would be smooth circumferential lamellae over the entire surface, we start to form these ridges. Now again, I'm going to draw it here to remind us. Remember, the periosteum is still on the outer surface of this, containing the immature cells, which is allowing it to divide. And that also means that that periosteum is also lining these ridges that are forming as well. Now, I need to add more layers of matrix, so I'm going to cheat and get rid of these, but I just put it there as a reminder. And these ridges continue to form. Oops, wrong color. Now, what ends up happening is that these periosteal ridges, as it moves beyond the blood vessel, will continue to expand. And eventually, what'll happen is the periosteal ridges will actually fuse together from the two sides. And as they fuse together from the two sides, again, remember, our periosteum is still on the outer surface of these. Actually, let's do this. And then it comes in here like that. And it comes in here like that. It basically pinches off this little piece of periosteum. Now, of course, there's still periosteum on the outer surface. So on the outer surface, we can continue to add more layers of matrix. And again, now it'll be a continuous layer of matrix. So our circumferential lamellae will continue to be formed. But now notice in here, we now have a small piece of the periosteum surrounding the blood vessel. Now notice, it's no longer on the outer surface. So should we really be calling it a periosteum anymore?
No.
Yeah, technically it's now an endosteum. But even though technically it's an endosteum, has the anatomy of it changed at all?
No.
No, it still is a dense irregular connective tissue, and it still houses mesenchymal cells. And of course, those mesenchymal cells can divide to form osteogenic cells. And those osteogenic cells can form osteoblasts. And those osteoblasts will surround themselves with matrix, forming osteocytes. And so as a result of that, I don't know how easy this is going to be to do. Yeah, I didn't think that was going to work. Well, maybe. Hey. I'm cheating a little bit, but heck, since we're online. Good enough. As a result of that, what's going to end up happening is we are now going to draw a layer of matrix or build a layer of matrix around this blood vessel. And because there's still an endosteum in here, we can draw another layer of matrix around this blood vessel. And now let's totally cheat and just get rid of my endosteum. And I can draw another layer of matrix around my blood vessel and another layer of matrix around my blood vessel. And suddenly, what have I formed here?
An osteum.
Yes, exactly. So with this endosteum, from the endosteum, I can form now concentric lamellae around the blood vessel. And I can continue this until I have formed an osteon. And lo and behold, not only have I made my bone wider, but I've also formed an osteon. I've also formed not just more bone on the outer surface, but compact bone. So this allows me to make the bone wider and make compact bone. Now, there is one more step to this process. We start with our baby bones with that medullary cavity in the center. And as we grow and as we make our bones wider and make them larger, does that medullary cavity stay the same size as it was before? Does all of our medullary cavities the same size as it was when we were babies?
No.
No, of course not. So of course, what we need to do is be able to expand it. I need a way to be able to make this medullary cavity bigger. And does such a thing exist?
Osteoclasts.
There you go, absolutely. Here in the endosteum, of the medullary cavity are osteoclasts. Remove matrix to enlarge the medullary cavity. So that not only does our bone get wider, but we also have an appropriate sized medullary cavity for it as well. And that is how our bones get wider.
Yeah,
And-
it's the same process. So sorry, good.
No, I was gonna say, not only do we make it wider, but this is also how we make our compact bone. Now, what were you asking?
I was gonna say it's the same process, just for the interstitial lamellae. Just basically just growing in between two different blood vessels.
Well, so I like where you're thinking, but notice in this case, we've made one osteon. But what if there had been a second blood vessel right here, right next to it? In that case, and I'll just cheat to make this a little easier, I'll use a big solid red. Notice if a second osteon had formed right there, then these little layers of irregular matrix in between that had been forming in between would basically then become the interstitial lamellae. So this disruption of the pattern is basically what forms the interstitial lamellae, and that's why they don't have a regular shape, because they're basically filling in the spaces in between the osteons. So just whatever grows there, filling in the space between the osteons, becomes the interstitial lamellae. Excellent. All righty, we've done it on the board. Let's take a look with the pretty pictures. Notice one other thing. There's one real, again, my drawing skills are limited, as you figured out. Notice here, though, we have our periosteum, we have our immature cells that are forming. And what's nice about this is we have both 2 blood vessels that are going to be incorporated into osteons. But notice these are interconnected. and this one is going to another blood vessel someplace else. So notice, not only is this going to be how we form the osteons and the central canals, this will also be how we form our perforating canals that connect the two blood vessels together. So, as we see, our periosteum has mesenchymal cells that are dividing to produce osteogenic cells. Osteogenic cells become osteoblasts. Osteoblasts make matrix. And we add a layer of matrix. They mature into osteocytes on the outer surface, forming those circumferential lamellae. All right, let's write that here. However, we have to respect the blood vessels. So as those layers of matrix are being formed, we have to respect the blood vessels, and so those periosteal ridges start to form around those blood vessels, giving us a more irregular shape to our outer layer. This process continues until our periosteal ridges fuse together. Notice again, great picture from your textbook of how a little bit of that periosteum is going to get pinched off into this space surrounding the blood vessel. Where now technically, because it's not on the outer surface, it is considered an endosteum, but it still has the fibers, it still has the mesenchymal cells, it still has everything it had before. So while we continue to add more layers on the outside, we can now start forming layers around that blood vessel until it's completely surrounded the blood vessel with concentric lamellae, right? Again, as it grows inward towards the endosteum, we are now forming concentric lamellae. And as we form those concentric lamellae, we ultimately form an osteon. And this process continues. Questions on that?
This is kind of random, but does there have to be a certain number of rings in order for it to be considered an osteon?
That's a great question. I would say no. I would say the key characteristic to an osteon is that it has that central canal with a blood vessel at the center, right? If you remember, when we saw the trabeculae, there are going to be some trabeculae that are just going to be made up of 3 or 4 layers of matrix kind of on top of each other. But if they don't have that central canal at the center, then it is not considered an osteon. So if you just had 3 layers of lamellae that were wrapped around each other, heck, you could have 15 layers of matrix all wrapped around each other. They still would not be considered concentric lamellae unless there was a central canal at the center. So I think the real key to the definition of an osteon is that central canal with a blood vessel at the center of it. All right. Great question. Any others? All right. Excellent.
And then,
Go ahead.
oh, it's the same principle with it stopping. Obviously, you don't want your bones to just continue to get fatter, fatter, fatter. Is it the same sex hormones that basically tell it to stop?
Great question. Obviously, it doesn't close. The periosteum never goes away. So you're right. It is going to be chemical signals that eventually will stop the outward width growth of our bones. It's a little bit more of a complicated process, but you've got the right idea. Absolutely, changes in chemical signals will stop the growth in width. Yep. All righty. Excellent. So there you go. Just that easily, we have now made our bones and we have now grown our bones. However, as we've talked about, once your bones Once you've made the bones and they've reached the term size, we're not done with them yet. As we talked about, bone is very dynamic tissue. Constant remodeling is taking place. We are constantly breaking down old bone, and it says tissue here. Really, it probably should say matrix. We're breaking down old bone matrix and adding new bone matrix to the tissues. And these are two important processes. The first is called bone resorption. Bone resorption is the process by which we break down old bone matrix. And of course, the cells responsible for that are the osteoclasts. Excuse me?
Coffee break.
Yeah, almost. And the second process is bone deposition. Deposition is the process by which we make new bone matrix. And of course, the ones that are responsible for making new matrix are the osteoblasts. Now, if our goal is to maintain homeostasis, what should the relative rates of resorption be in relation to the relative rates of deposition?
1 to 1.
Yeah. They should be equal. So as rapidly as we are breaking down bone matrix, we should be just as rapidly making it. So if we're breaking down bone matrix and making it at the same time, then our relative bone density stays the same, right? And we are at homeostasis and everybody is happy. However, is that necessarily going to be the case for our entire lives?
No.
No. When might it be different?
For women, it's menopause.
Okay. Well, so right. When we are elderly, and again, you are correct, this is more of a concern for females, but can it be a concern for males as well?
Yes.
Yes.
Absolutely. What is the relative rates then? What is the rate of resorption to deposition in an elderly person? Are they equal then?
No.
No. What is it then? Is resorption a higher rate? Yeah, resorption is greater. There you go. Resorption is going to be greater. Again, that's what we talked about. Grandma and grandpa, that for that matter, can be drinking a whole cow's worth of milk. But if our osteoclasts are breaking down bones faster than our osteoblasts are making them, then the bones are going to lose density and are going to get weaker. Now, that doesn't mean grandma shouldn't be drinking that cow's worth of milk, because if she's not getting that cow's worth of milk and her osteoclasts are more active, then that's going to cause even more problems. Would there be any other time in our life where these two processes would be different?
Puberty
There-
switched.
Well, not even necessarily puberty, but puberty is a good guess, but even before that, right? While there is a huge growth spurt in puberty, what about when you're an embryo or you're a fetus or you're an infant, right? At those points, the growth rates are even greater. So when we are very young, then again, in this case, when we're talking about the rates of resorption to the rates of deposition, right, then obviously deposition is a much greater process during that period of time, right? As we, because we talked about that baby bounces. So we need to convert a large amount of those connective tissues into bone, right? That baby rolls off the changing table, and even if it gets injured, right, by lunchtime, that bone is practically healed, All right, if grandma rolls off the changing table, has her hip regrown by lunchtime? No, her healing from that damage is much, much longer process because, again, resorption tends to happen faster than deposition. So absolutely, as our age changes, the rates of these change as well. Now, as we also hinted at, other factors can influence this. So when it comes to maintaining our bones and maintaining healthy bones, there are three main factors that can influence and affect our bone homeostasis. And what are those three main factors?
Hormones.
Excellent. Hormones, what hormones are produced are definitely one of the things that can influence it. What's
Nutrients.
another? Yeah, we talked about grandma drinking the whole cow's worth of milk, nutrients, and what's the third?
Exercise.
There you go. So when it comes to maintaining our bones, there are three main factors: hormones, nutrition, and exercise. And so that is what we are going to talk about next. So next we need to talk about these three characteristics: hormones, nutrition, and exercise, and how they help us to maintain homeostasis. But we'll go ahead and save that for after our first break. It is 9:02 right now, so let us restart at 9:17, and I will start the recording at that point. Any questions before we take our first break? All right, excellent. I will see you guys in 15 minutes. All righty. Let's go ahead and pick up where we left off. Our goal now is to maintain bone homeostasis, and let's start with some of the easier ones first. Starting first with exercise. Activity is vital for bone maintenance. However, when I say activity, does that mean that all exercises affect the bones equally?
No.
No.
No. To maintain the health and strength of the bones, what kind of activity is actually required?
Weight bearing.
Exactly. With this, what we really want is some type of weight-bearing activity. And the reason for this is that it puts stress on the bones. Stress in this case is actually a really good thing. Without that weight-bearing activity, without that ability to put stress on our bones, we can lose as much as 1/3 of our bone mass in weeks. Right? Obviously, this is a major concern for our astronauts who are in outer space without a gravity. So they have very elaborate and specialized types of Stairmasters and other types of exercise machines that they use to help to put stress on the bones. But it also can affect you and I. You hurt your back, if you break your leg or do something like that where you're laid up in bed for a week or two, that can have a significant impact on the stress that we're able to put on the bone and therefore the strength of the bone. The reason for this is that stress basically influences and strengthens the bone. It makes the bone stronger, and it makes the bone stronger in two ways. The first way is it increases the density of the bone. One of the classic places where they've seen this is, for instance, in tennis players. Right, tennis players, sure, they make plenty of shots two-handed, but obviously their serves are one-handed, many of their shots are one-handed, and so they clearly use one arm more than the other. And they've actually taken x-rays of the arms of tennis players, and they find that the dominant arm has a much thicker and much stronger bone than the one that is their non-dominant arm. And the reason for that is that stress that they're putting on it. It makes the bones thicker, but it also makes it stronger. And the way it makes it stronger is that it aligns the osteons. Our osteons, remember, our bone is dynamic tissue, constantly being broken down, constantly being rebuilt. And when we rebuild our osteons, we do that to align them across the stress, right? The same way that we have those collagen fibers in a dense regular connective tissue that align to provide the stretch and the strength for a ligament, our bones align along stress as well. This is important because it helps to strengthen the bones for the activities that we're participating in. But again, it's one of those things that can be a concern as well. If you have a chronic injury or some other long type of lasting injury that causes you to change your gait, changes the way that you move your body, that can actually cause, as you're shifting your weight, that can actually cause a misalignment of your osteons. So they're not aligned along the same lines as they were before. You see this sometimes in professional athletes. A professional athlete will have some type of soft tissue injury where they have a groin strain or they have an Achilles tendon injury or something along those lines. And so it changes the way they move their body through space. And sometimes when they come back from that soft tissue injury, suddenly they break a bone. And that can happen because they've weakened the bone by misaligning it, by changing the way that their gait is located. The other way that you see this strengthening of the bone from the stress is that when the muscle pulls on the bone, it increases the attachment point. I made the joke about the bump on the back of your head. Remember, we talked about the bump on the back of your head, which we now know is the external occipital protuberance. And I made the joke that if you have a big bump there, that means you fell asleep a lot in class, because when you fell asleep a lot in class, that ligamentum notia, that elastic ligament, had to pull your head back up, right? So I made that joke. If you're having to have to pull your head back up because you fall asleep a lot, that gets bigger. Although where are the places you see it the most is in the legs, right? We haven't talked about the leg bones yet, but we have talked about the Quadricep Femoris, which is this big, huge, large muscle group in the leg region, your femoral region of your body. It's the largest muscle group in the body. And as we talked about, it attaches to a tendon to your tibial tuberosity, a bump on the anterior part of your tibial bone, basically the top of your, what we commonly think of as the shin. For bicyclists, for soccer players, for distance runners, often they will have a very pronounced bump. That tibial tuberosity becomes quite enlarged because as they're constantly running or they're constantly riding their bike, that muscle is constantly pulling on that attachment point, and that attachment point gets larger. Right? Excellent. Now, the question becomes why? What is it about this stress that causes the bones to get both stronger and more aligned and increase the size of their attachment points? Well, how many people have ever eaten a wintergreen lifesaver in the bathroom with the lights off?
Hey,
You? Excellent. Why?
sparks.
Because you just, exactly, right? I know it sounds like an odd thing that you would want to do. Excellent. A couple of you have done it, and Brian hit it on the head. If you've ever been like my daughter at her Girl Scouts, both they did that when they went camping, or you've ever had a sleepover or something like that. If you haven't, then that's your assignment this weekend. Your assignment this weekend is to either go to the store, or again, if you're still not going to the store, then DoorDash or whatever, some wintergreen lifesavers. Get into the bathroom, turn off all the lights, pop one into your mouth, and then rapidly chew it. And when you do that, as Brian mentioned, what happens? What did you see when you did that in the mirror?
Like, looks like it sparks. Like,
you get sparks, exactly. Because what happens is when you put the pressure of rapidly chomping on those crystals, it actually produces a small electrical current. And you will see little sparks in your mouth. And that's exactly what happens with our bone matrix. When we put pressure on those hydroxyapatite crystals, those hydroxyapatite crystals produce a little electrical signal. And that little electrical signal stimulates the osteoblasts. And so those osteoblasts build bone faster. Yes, you get the advantage at the sleepaway camp of being able to chew on those, and everybody goes and ahs at the sparks that you're producing. But like many things, once we learn about the body, we also learn how to use these things to manipulate the body to our advantage. One of the things that we've talked about is that bones are very well vascularized, and because of that, they heal very rapidly. But like we've always said in this class, never say always, never say never, right? Most bones. There are a few bones that aren't very well vascularized, right? There happen to be a couple in your ankle that don't have a good blood supply. There's a couple in your wrist and your patella, right? Patella is a sesamoid bone sitting in the middle of a tendon. And as we know, tendons don't have a good blood supply. So your patella doesn't have a good blood supply either. So let's say, for not so theoretical argument's sake, you happen to be the greatest receiver of all times. All right, starting out on a total loser team like the 49ers, but finally get smart, end up on the Raiders before they abandoned us and went to Las Vegas. Screw them. But you spent a brief period of time there, Jerry Rice, and Jerry Rice actually broke his patella one day during a class. And one of the things that they did is they actually took some electrodes and inserted those electrodes into his kneecap to stimulate the bone and cause it to heal more rapidly. They do that sometimes in the wrist or sometimes in the ankle. So some of these bones that are poorly vascularized and normally heal very slowly because they don't have a good blood supply, by inserting electrodes and providing an electrical stimulus, we can actually stimulate those osteoblasts and grow that bone back more readily, more rapidly. So again, as we'll see again and again in this class, as we learn how the body works, we use that to manipulate it to our advantage. And this is a great example of that. All right. Questions?
Is that kind of like the epidural, I'm probably going to say it wrong, but epidural growth? We were talking about like when you do like that and it makes it grow faster.
True. Okay. So yes.
Similar, but not the same.
So it's similar in that in both cases, you have cells that are more active. In the case of the skin, it's a chemical signal, that epidermal growth factor, that is stimulating them to grow faster. In this case, it's actually the electrical current that is stimulating the osteoblast. So you're right in that both cells are being stimulated to grow faster, but the stimulus is very different between the two. But you absolutely have the right idea. All right. I think of our three characteristics, that's exercise. That's the easiest to understand. Any other questions on this one? All right, similarly easy, but again, like most things, a little bit more complicated, is our nutrition. Right? Obviously, there are certain components that we need within our diet to be able to maintain healthy bones. And the most obvious of those are calcium and phosphates. As we mentioned, our bone matrix is comprised of those hydroxy, apatite crystals, right? They are the strongest component made by the human body, and they're primarily made out of calcium phosphate and calcium carbonate, and there are additional things like magnesium, manganese, fluoride, iron, things along those lines that are required to make the matrix. So we require daily intake of these micronutrients to be able to maintain our bone density, to maintain that bone strength. But it's more than just the matrix itself that is needed for strong, healthy bones. For instance, we also talked about vitamin D. Where did we say we can make vitamin D again?
In the skin.
Mm-hmm. Remember, in our skin, we have a special cholesterol that when exposed to UV radiation gets converted into vitamin D. However, as we also talked about, everybody's hiding inside these days. So luckily they put vitamin D in a lot of food that we have. Well, when vitamin D is activated, it becomes a hormone called calcitriol. Calcitriol, of course, is a chemical signal that has the potential to target every cell in the body. Of course, most hormones don't target every cell in the body, and in particular, calcitriol targets the digestive system. And it targets the digestive system to tell our digestive system to absorb calcium from our food and drink. So again, remember, we know the lumen of our digestive system is outside of the body. So if we need to get it inside of the body, we need to pass the membrane. And calcitriol tells the digestive system, hey, you see any calcium laying around inside that lumen of yours, bring it into the body. We need it. So obviously, you can drink a whole cow's worth of milk. If you don't have that vitamin D, you're not going to be able to absorb it into your body. But also vitamin C is vitally important. Right? Back in ancient times, it was once said that the sun never sets on the British Empire. And they said that because basically Britain had colonies around the entire circumference of the globe. And what was it that allowed the British Empire to be so dominant? Control the whole world?
Ships.
Yeah, they had the most dominant navy of any country on the planet. Now, as dominant as that navy was, it was almost completely devastated by this horrible disease where the sailors' bones were breaking and their gums were bleeding and they were losing their teeth and their skin was peeling off of their body. What was this disease called?
Scurvy.
Scurvy, absolutely. Right? And what was the miracle cure they came up with to be able to combat scurvy and allow the British Empire to at least last for another couple hundred years?
Fruit, citrus.
Yeah, exactly. Limes. Exactly. It turns out what was happening, these sailors who were on boats for long periods of time didn't get vitamin C. And it turns out vitamin C is vital for making collagen. As we know, collagen is the most common protein in the body. So again, it helps to hold our skin together, helps to hold our teeth in place. That's why they were losing their gums. I mean, their gums were bleeding, they were losing their teeth, their skin was tearing. But remember, we also talked about in the process of deposition, our osteoblasts are taking that calcium, taking those salts, and depositing them on collagen fibers. So if you don't have collagen fibers, you can't make hydroxyapatite crystals. And so their bones were weakening and breaking as well. So again, vitamin C doesn't help us make the matrix, it helps us make the collagen fibers. And without the collagen fibers, we have no place to put those matrices. And there are other micronutrients as well. Vitamin A, vitamin B12, vitamin K, they all play an important role in helping to make and maintain our bone marrow. Notice bone marrow doesn't necessarily directly relate to the strength of our bones, but why is it important to make and maintain bone marrow, especially red bone marrow?
Used in the production of red blood cells.
Yeah, and not just red blood cells, but all of our blood cells. But you're right, red blood cells are one of the big ones. As we know, the red blood cells carry about just under 98% of the oxygen in our body. So if a cell needs oxygen, it's that red blood cell that's going to provide it. And as we've also talked about, red blood cells are anuclear. They're just bags of hemoglobin, and they only last about 100 days. So every day, millions of red blood cells are dying in your body, and you need to replace those millions of red blood cells. So having these micronutrients are vitally important for maintaining our bone marrow, maintaining our bone health, and our overall health as well. Excellent. So this is some examples of the way that our diet influences and affects the homeostasis of our bones. Questions on that? All right, excellent. Here are where things get a little trickier. Hormones, right? Let's go to the whiteboard for this part. Right? Obviously, to maintain healthy bones, we have to have hormones that help us to regulate bone density. But as we've also talked about, that bone matrix is primarily calcium. And so those same hormones that help us to regulate bone density also can play a role in regulating calcium levels in the blood. And as we've talked about, maintaining calcium levels in the blood have the potential to be detrimental to our bone density. Now, let's think about the two processes we've talked about: resorption and deposition. Those are the two that ideally we want to maintain in homeostasis. Resorption is when we do what? Make matrix or break down matrix?
Breakdown matrix.
Excellent. So when we break down matrix, we do two things. Obviously, we decrease the density of our bones. But when we break down that matrix, where does the matrix go?
Into our blood.
Yeah, the calcium enters the interstitial fluid, right? But from the interstitial fluid, that interstitial fluid freely mixes with the blood. So really what it is doing, it is add calcium to our blood. And so it increases calcium levels in our blood. Conversely, deposition is when we make new matrix. That obviously increases bone density. And of course, where does it get the calcium to make that new matrix?
From the blood.
Right. It again removes the calcium from, again, directly from the interstitial fluid. But of course, where did the interstitial fluid get it from? It gets it from the blood. And that would lower calcium levels. So when we think about the hormones, we need to think about the effect, not just that they have on the bones, but we also need to see if they have an effect on calcium levels in our blood as well. Now, let's think of these in terms of some of the hormones we have already talked about. The first hormone we just finished talking about was vitamin D. Right? Calcitriol, sorry. Calcitriol is the active form of vitamin D. As we know, it comes from either the skin or diet. And what does calcitriol tell us to do again?
Absorb calcium.
Yeah, it targets our digestive system to absorb calcium. So what's the direct effect on that?
It's going to increase the calcium in our blood.
Excellent. It increases the calcium in our blood. Now, directly, does that affect our bone density?
No.
No. But as we know, if there is more calcium in our blood, then there's more calcium available to make bone. But it doesn't directly affect that. So notice this first hormone we talked about really just affects calcium levels, doesn't have a direct effect on the bone. However, we did talk about hormones that have a direct effect on the bone. Sex hormones. And what were the sex hormones again? The classes?
Estrogens and androgens.
And androgens. Excellent. What was the target of our sex hormones? What did our sex hormones influence? Excellent. The target was the osteoblasts. And what effect did they have on the osteoblasts?
Increase production.
Yeah, they stimulated them or excited them. And when you stimulate the osteoblast, the effect of that is what?
Removing calcium from the interstitial fluid, the blood.
True, absolutely. So this is going to lower calcium levels in the blood. And is there another effect of that?
Increased density in our bones.
Yeah. Excellent. So it increases bone density, and of course that lowers calcium levels in our blood. And one more thing. Do both estrogens and androgens have, are they equal in their effect?
No.
Bingo. Excellent. So notice right here, right off the bat, we've talked about two hormones that we've already talked about in class. We're just kind of putting it into this relationship of the hormonal regulation. And that, again, really isn't new information. We are just kind of consolidating and organizing the information we've already talked about. But let's talk about some other hormones that we haven't talked about yet. The first one is calcitonin. Notice similar sounding to calcitriol, but it is not the same thing, calcitonin. Calcitonin is actually produced in the thyroid gland. And it turns out it targets the osteoclasts. Now, in this case with the osteoclasts, what it actually does to the osteoclasts is it inhibits them. It reduces their activity. Right? But it also has other targets as well. The second target is the kidney. I think we mentioned this in the class. Your kidney filters 200 liters of fluid out of your blood every single day. Now, does it release 200 liters of fluid from your body every day?
No.
No, you don't have 200 liters of extra fluid you could get rid of. And also, if you produce 200 liters of urine during the course of the day, we'd all be sitting in the bathroom right now while we were doing this lecture. So while clearly a lot of stuff comes out, it also reabsorbs a massive amount of stuff. However, in this case, calcitonin tells the kidneys, you know what? Don't reabsorb calcium. Any calcium that sneaks its way out into that filtrate, it's okay to go ahead and let that leave in the urine. And there is a third target. for calcitonin. And that third target for calcitonin is actually calcitriol production. It basically tells the body to stop making calcitriol. And if we have a drop in our calcitriol, what's going to happen to our calcium reabsorption from our digestive system?
Decrease.
Yeah. Excellent. So 3 main targets of our hormone calcitonin. Now, what are the direct effects of this? Let's look first at the first effect. We are inhibiting the osteoclasts, right? Normally, we're breaking down bone at the same rate that we're making bone, right? They're doing it at the same time, and our bone density stays the same. But if we keep making bone at the same rate, but we slow down the breakdown of the bone, so now we're slowing down the breakdown of our bone, but we're still making it at the same rate. I'm not coordinated enough to do that at the same time. What's going to end up happening to our bone density?
Increase.
Increase.
Yeah. So our bone density increases as a result of this. Of course, where are we getting that calcium to make that bone density?
From the blood.
From the blood. And if we're telling the kidneys to let calcium leave the blood, and if we're telling the digestive system to stop bringing calcium into the blood, what effect is this going to have on blood calcium levels?
Decrease.
Blood calcium levels drop. Excellent. So notice calcitonin here both increases our bone density, but it also drops blood calcium levels in our blood. Right. So again, you have that nice big huge glass of milk. Calcium levels in your blood go up. Calcitonin is going to tell our bones, stick that into the bones. Let's get rid of the rest. Let's stop bringing as much in, and it's going to bring calcium levels back down. But what happens if you have a Diet Coke and a donut for breakfast? Are you necessarily getting the calcium levels you need then? No. And so luckily we have yet another hormone called parathyroid hormone. And with a name like parathyroid hormone, I'm guessing you can guess where it is produced.
Thyroid.
How about in the parathyroid gland? When we get to the endocrine system, we'll learn that the thyroid gland is this kind of C-shaped gland that sits underneath your Adam's apple. And on the back of it are these small little globular glands that are actually separate glands, but since they're attached to the thyroid, they're called parathyroid glands because para means alongside. So parathyroid hormone is produced by the parathyroid gland. And guess what? It happens to also have 3 targets. Target 1 happens to be osteoclasts. However, in this case, it excites or increases the activity of the osteoclasts. It targets the kidney. And it tells the kidney, hey, you got any calcium in that filtrate? Go ahead and bring that back. Absorption, so less calcium leaves in the urine and more is brought back into the blood. And, not surprisingly, it targets calcitriol production. Increasing calcitriol production. Right? And that basically causes more calcium to be absorbed from the digestive system. So, what are our direct effects here?
Decreases bone density.
Well, is this going to increase or decrease bone density?
Decrease. Bone
Decrease,
density.
right? Because notice, we're building bone at the same rate that we were before, but now we're breaking it down much faster. Our osteoclasts have now been stimulated. So we're breaking it down a lot faster than we're making it. So the net effect of that is to cause a decrease in bone density. Right? But by releasing that calcium from the bone into the blood, by bringing more back from the kidney, by bringing more in from the digestive system, what it does do is increase blood calcium levels. So it brings calcium levels up. Notice in this case, as we've talked about, we're actually damaging the bones to make sure we have the appropriate amount of calcium in our blood, because having the appropriate amount of calcium in the blood is vitally important. I'll show you how important in just 1 minute. So there you go. 4 hormones, really 5, but 2 of them, estrogens and androgens, basically do the same thing, that all have the potential to both affect our bones and also affect calcium levels in our blood. We've done it here with all the pretty words. Let's take a look at the pretty pictures. Again, as we've talked about, our sex hormones stimulate the osteoblasts, and estrogens do that more. Calcitonin, remember we talked about from the thyroid gland, is going to decrease blood calcium levels. It does that by inhibiting the osteoclast, so we make more bones. But also, oh, why does that say that there? Also targets calcitonin in kidney, so we talked about that. And parathyroid hormone also influences those same 3 targets. Stimulates the osteoclasts, here we go, tell the kidneys to reabsorb calcium, and increases calcitonin. So let's put that there as well. Again, I may not have it all here in the words, but we've done that. But I love this picture from your textbook. Notice here, we see what happens when calcium drops below the normal range. And in this case, notice the normal range is 8.5 milligrams per deciliter. If it drops below, we have to bring it back up. How do we do that? Primarily parathyroid hormone. Parathyroid hormone tells the kidneys to bring more calcium back. Parathyroid hormone stimulates the osteoclasts, releasing calcium from the bone into the blood, and parathyroid hormone stimulates the production of calcitriol, so we absorb calcium from our digestive system. Conversely, if calcium levels get too high, and notice too high is 11 milligrams per deciliter, we are going to produce calcitonin. Calcitonin tells the kidneys, hey, all this extra calcium in our blood, get rid of it. Let it go in the urine, right? By inhibiting the osteoclasts, more calcium is going to be deposited into the blood by the osteoblasts, not because the osteoblasts are stimulated, but because we're not breaking down matrix as quickly. And we stop calcitriol production, so we stop the reabsorption of calcium from our digestive system. Now, you've been in class with me for, what is this, our third week now? If you've noticed, I'm not a huge numbers guy, right? There aren't too many numbers you've had to memorize. Eventually, there'll be some numbers that are going to be important that you'll have to memorize. These aren't some of them, right? Now, again, knowing that the normal range of calcium in your blood is 8.5 to 11 milligrams per deciliter is important information because it's the kind of thing that you can use to woo women in a bar. Right, all you have to do is go into a bar now that where bars are open again, walk up to a woman, saddle up next to her and go, hey, did you know that the normal levels of calcium in your blood are between 8.5 and 11 milligrams per deciliter? And they just swoon when you do that, right? So again, it has its uses, that kind of information. These are not numbers you have to memorize, though. The things that can easily be looked up. But why I make a point of emphasizing them here is look at how narrow this range is. 8.5 to 11 milligrams per deciliter is an incredibly small range. And so the only reason I point it out to you now is, again, to really emphasize to you how important calcium is to the function of the body. Calcium makes cells do wonky things. And so we need to very, very carefully control calcium. If we want a muscle to contract, if we want a neuron to send a nervous signal, if we want our sweat glands to release sweat, many of the functions of our body are controlled in some way or influenced in some way by calcium. So it's not surprising that we have to keep such a tight control of calcium in the body. And that's why when it comes to either damaging our bones or making sure we have the right amount of calcium, the right amount of calcium is going to win every time. All right. Questions on that? All right. Excellent. So with that, we have now talked about the three main factors that we need to maintain our bones: exercise, nutrition, and hormones. And not only have we talked about the hormones that help us to maintain the bones, but how those hormones help us to maintain calcium levels as well. What that means is we are done with all of the normal physiology we need to talk about for bones. So all we have left, what we will do, today's Friday, so I guess we'll do it Monday, is we will now talk about how to break and heal our bones, our major homeostatic imbalance to our bones, and then the healing from that. And then from there, we get to go into joints. Now, what I want to emphasize, especially after what I've seen so far on the exams, I stayed up very late last night trying to get through the exams. All of the lab exams are graded, although I have not put in the curve and modified the grades that way. And I'm halfway through the lecture exams. And a couple things have stood out very, very clearly to me on these exams. The first, that again, like we talked about in the last class, despite my warnings, people are not answering all parts of a question. A lot of people forgot right and left and things along those lines. But the other thing that I noticed is for most people's lab exams, they did a lot better on the earlier parts and a lot worse on the skin part, especially the glands. Now, granted, that was the things that we talked about on the last day, so you had the least amount of time to study that. But I will remind you that you know the things I'm going to talk about. You have the lecture outlines, and even if the lecture outlines don't make sense to you, when you see, for instance, that we're going to be talking about joints next week, there's nothing to say that you can't go to the textbook and read them. One of the things that I found is typically the joints are the part that people have the most problem with on this exam. Not because I think they're necessarily harder, but because what I see about the joints is it's the terminology. It's the vocabulary. You have things like synchondroses and syndesmoses and amphiarthroses, right? And all of these, gumphosis, and all of these elaborate terms. Your book does a nice job of describing and organizing these. There is a great handout I guess I would say study guide, but really it's just a handout of organization for the joints. It's not a homework assignment, but it does a nice job of organizing our joints, because when we talk about our joints, you need to talk, like many things we've talked about, like the glands, which you guys struggled with. There are functional classifications to our glands. And I'll tell you right now, there are 3 of those. There are structural classifications. And I will tell you right now, there are 4 of those. But each of our structural classifications have specific types. And that will be the vocabulary I use. So on the exam, if I point at a particular joint, why am I doing that? So here are 2 bones that are coming together to form a joint. I could point at this joint on the exam, and I can ask you one of 3 questions. Identify the functional classification of this joint, identify the structural classification of this joint, and identify the specific type of joint. So 3 different questions with 3 different answers. Obviously, they're related to each other, but this is information that you need to organize and be comfortable with. Your book does a really good job in this the joints chapter of talking about these things, but there's also that really great handout online in our module that really distills the information down, shows how these things relate to each other. So make sure you're studying this. Remember, this is your big 2-day weekend to study this material. All right, how are we on time? We are early, so let's do this. Before we take our next break, because then during our next break, I want to put you guys into your groups again just to get started with that. Let's go ahead and switch gears from here back into our skull features that we were talking about. So if you need a minute or 2 to shuffle your papers, do that. We left off last time, and we had just finished off the orbit of our eye. So again, remember, as a reminder, when we're talking about our skull features, on the exam, there are 2 questions I could ask you. One, of course, is to identify the skull feature. And of course, what skull feature are we looking at right here?
The orbit.
Yeah, the orbit of the eye, exactly. Or the orbit of the skull, right? Absolutely. Excellent. Or remember, question 2 I could ask is to identify the bones and bone features. However, for the orbit of the eye, are there any bone features we have to worry about?
No.
No. So in this case, the second possible question I could ask would be identify the bones that form this bone, this skull feature. And what would the answer to that be?
Frontal, maxillary, zygotic, ethmoid, sphenoid, palatine.
And lacrimal.
There we go. Excellent. 7 bones. And again, I already told you my cheat for this. My cheat, my mnemonic is to remember the north, the south, the east, and the west, right? The frontal up here at the top, maxillary here at the bottom, zygomatic here lateral, lacrimal here medial. And if I can remember that, then I can remember what's on the inside, because I happen to have a little ESP, right? After all, if aliens are real, then anything can be real these days. So again, our government is now telling us that aliens exist, or at least UFOs do. And so then why can't ESP exist as well? All right, so there you go. All you need is a little ESP, and then you can remember those. All right, questions on that? All right, that brings us to our next skull feature. And if I remember correctly, our next skull feature is the jugular foramen. Is that correct? Now remember, skull features are made by 2 or more bones coming together. Remember, we talked about our zygomatic arch. being made of the zygomatic process of the temporal bone and the temporal process of the zygomatic bone. And those two processes come together to form our zygomatic arch. But the other way we can make a skull feature is by having a bone that has an indentation in it. That indentation by itself isn't anything more than an indentation. But if we happen to have a bone next to it that also has an indentation, then lo and behold, we have a hole in the skull. And that's exactly what happens here. Notice there is a notch in the occipital bone. There is a notch in the temporal bone. And when those two things come together, they form the skull feature that is the jugular foramen. And again, while we don't need to know functions, guess what one of the things that passes through the jugular foramen is?
Jugular.
Yeah, jugular vein, absolutely. Now notice one other thing. Notice, for instance, up here, when we look at the foramen ovale, or we look at the foramen spinosum, or we look at the carotid canal, notice those bone features typically are more smooth because they're a hole that is formed within a single bone. When we have the notch of one and the notch of another coming together, and let's go ahead and cheat and change colors to really emphasize this. Notice that when we have two notches that come together, they tend to be more irregular in shape. Much more bumpy, much more irregular. And in fact, notice here is another bumpy and irregular shaped opening. Anyone know what these two up here are called?
Carotid.
Not a bad
Canal.
guess, although remember the carotid canal tunnels through here. Notice these openings are so irregular and jagged, it almost looks like they're torn open. What might a good name for a torn looking opening be?
Fissure.
Not a bad guess because we need something bigger, but nope. Think more in terms of the torn. Torn, dare I say even maybe lacerated.
Foramen lacerum.
There you go. That is the foramen lacerum. Notice the foramen lacerum is formed by three bones coming together. Here, I'll emphasize it on this one. Notice here, we have three bones that are coming together. The occipital bone over here, the temporal bone over here, and then the sphenoid bone up here. These three bones kind of fuse together to form this very irregular shape opening known as the foramen lacerum. So here are two of our skull features we are responsible for. Now, in the best of circumstances, when we're sitting here in the classroom and you get to hold the bones in your hand, it can still be hard to tell the difference of all of these openings, right? When we're just looking at pictures, it's even worse. So the two things to really pay attention to are the relationship of these things to each other, and also the questions I ask you on the exam. Notice, again, when we're talking just about the holes that we're looking at here, they're basically going to be two types. There are going to be bone features. And of course, when you have a bone feature, you're also going to need to name the bone that it is, and the skull features. So, for instance, even if you know nothing else, if I put the question, if I have an arrow pointing at one of these openings, and notice I'm purposely not pointing at one of these openings, and I ask you to identify the bone feature, is the jugular foramen ever going to be the right answer to that?
No,
No,
Why not?
because it's a skull feature. It's
Exactly.
two bones coming together.
Exactly. And so that can help you on the exam. Notice if I have this arrow and I'm pointing it right here, and I ask you to identify the bone and bone feature, you know it's not the jugular foramen. You know it's not the foramen lacerum, because those are not bone features. As I said, I try to be very precise, not because I'm trying to be tricky, but because there's a lot of specific information. And the good news is you can use that to your advantage. If I ask for a bone feature, then that limits the number of things that you're responsible for. If I ask for a skull feature, that limits the things you are responsible for.
Professor?
Yes.
So would you, for the answer, would you be looking for the bones that make up the foramen lacerum?
So great question. So again, notice with, let's actually put that arrow back. If I put an arrow in something like that, there is really only one question I can ask. The one question I will ask is to identify the bone and bone feature. And let's make that red so that it reminds us of that. So with a bone and bone feature, that's the only question I can ask. However, if I put an arrow here, there are two questions I can ask. Question one is identify the skull feature. And question two is identify the bones that form. And again, it could be bones and bone features. Bone features wouldn't be appropriate in this case, but the bones that form the skull feature. So let's do that. Let's work our way through this, because like I said, one of the things that I find is that it can be challenging to learn this material, even under the best of circumstances. So let's start anteriorly and work our way back and identify all the holes. And we'll use the colors to make it easier. Identify the bone and bone feature.
Ethmoid bone and cribriform plate.
Okay, excellent. That would be correct, but remember we were talking about the holes in this case. So bone feature slash space. What is the bone feature slash space that is in that cribriform plate?
Olfactory foramen.
There you go.
Yeah, that one is the one that's confusing the hell out of me. That one and the cribriform plate, they're just,
So
for some reason.
think of it this way. If you have a cheese grater, you have the flat surface of the cheese grater and you have the holes in it, right? The cribriform plate is the flat part. The olfactory foramen are the holes. And that's what you have.
Yeah, thank you. That made it easy.
So yeah, so you're correct. In this case, my question was confusing. So I would probably, like I just mentioned there, I would say bone feature slash space. So you know I'm talking about the holes and not the flat part. Okay, excellent. All right, identify this bone and bone feature.
The sphenoid bone and the optic canal.
Excellent. It's a little tricky to see, but there's that tiny little hole here projecting anteriorly. Any idea what that is?
Is that the foramina spinosum?
No,
This one is the round one that points forward, so rotundum.
Rotundum.
And what bone is that?
Sphenoid.
Excellent. I'm not going to do the superior orbital fissure because we can't see it from this point of view. So instead, what about this nice oval shape one right here? I guess I can't do ovals, so we'll cheat and do that there. What is this oval shape one right
The
here?
jugular foramen.
But again, this is
Is
a
a
bone feature.
Oh.
sphenoid foramen ovale. Ovale?
The
Ovale?
sphenoid is again the bone, and you're right, foramen ovale, because it's oval shape. And next to the foramen ovale is this tiny little pinprick. What's this tiny little pinprick?
Sphenoid foramen spinosum.
Excellent. Spine, tiny little spine-like one. Excellent. So far, so good. We have to cheat a little bit for this one. There is the opening of a tunnel that we can see coming out into this space. What is that? Right, and the same thing over here. I guess I should be marking it over here since I'm writing it down here. What was this kind of tunnel-like that comes out from here that we can not see the best from up here?
Temporal carotid canal.
Excellent. And again, the big obvious one. What's that?
Foramen magnum.
And what bone is that?
Occipital.
Occipital.
Excellent. Identify the skull feature that the carotid canal points towards.
Foramen lacerum.
And what bones form this skull feature?
Occipital, temporal, sphenoid.
Excellent. And this skull feature, what is it?
The jugular foramen.
And what bones form it?
Occipital and temporal.
There you go. So get used to looking at these bones and notice part of the challenge, part of the problem with this is where things are located on the inside of the skull are slightly different from where things are located on the inside of the skull. Notice in particular, and I'm going to cheat on this one here by erasing that and that. Oops, nope, not that one. I want to erase that. Excellent. To more emphasize that we really don't see the opening of the carotid canal, but we know it comes out right here, basically right next to the foramen lacerum. I make a point of emphasizing that because notice when we clear this drawing and flip it over, whoops. Oh no, I haven't, there we go, an underside view. Notice here is the, oops, it needs to be green. Here is the foramen lacerum. Here is the jugular foramen. And notice here on the underside, this is where the opening of the carotid canal is. Notice on the underside of the skull, the carotid canal is right next to the jugular foramen. But on the inside, it's right next to the foramen lacerum because it tunnels through that bone. Foramen ovale, foramen spinosum. Notice we don't see the rotundum because it projects anteriorly, not inferiorly. But notice we see the stylomastoid foramen under here, still see the jugular canal. And as someone pointed out, even though we don't see it, we know the hypoglossal canal tunnels above those occipital condyles. So you have to get used to looking at the skull from different points of view to be able to recognize these openings. It's a lot easier when you can hold these things in your hand. The nice thing is you have some really good atlases, and that personal anatomy lab allows you to rotate the skull so you can look at it at different points of view to see how these things move through the skull. All right. Questions on that?
Yes. Would you mark us wrong if, like, let's say, we said magnum foramen, like if they were
No.
backwards,
If
or
you say foramen ovale or ovale foramen, well, technically one is true and one is not. The fact that you have both words, as long as you have both words, then I would give that to you. All right. Excellent. Now, if you said fissure ovale, that would be wrong. Well, you get partial credit. You get partial credit for the ovale, but you lose partial credit for the fissure instead of foramen. Okay. All right. What's left of our, I think it's just the indentations, the fossas, and then the sutures. Is that all that's left, or am I forgetting something? What's next on our study guide?
Inferior orbital fissure.
Ah, thank you. Okay, excellent, perfect. Here we go. Notice we've already talked about the superior orbital fissure. We know that is a bone feature between the wings of the sphenoid. But notice down here, there is also an inferior orbital fissure. This inferior orbital fissure, notice, is made by the sphenoid bone, the maxillary bone, and for many people, it will also come over here and involve the zygomatic bone as well. There are some people that it doesn't involve the zygomatic, so you can put zygomatic or not put zygomatic, I don't care. But definitely the sphenoid, definitely the maxillary bone. What's interesting is this opening is just as big as the superior, but there's really no major structures that pass through it. Lots of nerves and blood vessels pass through the superior orbital fissure, which is a bone feature. But our skull feature, the inferior orbital fissure, nothing major passes through it. There are some small blood vessels and stuff, but nothing major passes through it. All right, now are we in the fossas, or is there anything else? What's next on the list?
We did foramen lacerum, so yeah.
Last one. Okay, perfect, excellent. All right, so the last thing we need to do, second to last thing we need to do is these fossas. Notice when we look at the cranial cavity, the cranial cavity is basically divided into 3 indented regions. One indented region here, a second indented region here, and a third indented region here. Notice this third one is the deepest. This one is the most shallow, and there are basically 3. So not surprisingly, from front to back, what do you think we call these 3 indentations of the skull?
anterior,
Middle and posterior.
middle,
And middle and posterior what?
cranial fossa.
Cranial, because this is the brain. Fossa, which is an indentation. All right, so we have our anterior cranial fossa, middle cranial fossa, and posterior cranial. So how many fossas?
3.
3. And conveniently enough for us, 3 bones make each of them. What are the 3 bones that make up the anterior cranial fossa?
Frontal, ethmoid, sphenoid.
What are the 3 that make up the middle cranial fossa?
Phenoid. Temporal. Would you do the other temporal, left and right?
You are correct. There are indeed two, but I mean three different types.
Parietal?
Yeah. Notice above the temporal, forming the side, is the parietal. So there you go. Now you're right. There are also two of those as well, but I mean three types. So the phenoid, the temporal, and the parietal form those. And so what are the three that are going to form our posterior cranial fossa going to be?
Occipital.
Parietal?
Parietal.
Temporal.
There you go. Just that easy. Three indentations in the skull, three types of bones involved in making all of them. Yes, both temporals, yes, both parietals, but three types. It makes it easier. Again, I'm all about mnemonics and easy ways to remember those things, and those are the things that help me.
Would you ask a question of like three types or name all
No,
the bones?
I would ask you to identify the bones that form this skull feature. So you would be responsible for remembering that there's three. So I wouldn't ask for three. I would ask you for the bones that form this skull feature.
So then we'd write down 5, let's say for the middle.
Well, you could say the sphenoid, the two temporal, and the two parietal. That would be fine. Yeah.
Okay.
Yeah. All right. The last thing, and I'm going to cheat and go to the whiteboard for this, is to talk about these sutures. Sutures are joints, as we will learn and talk about in the next class. They are special joints, and they're special joints in two ways. They're non-moving, and more specifically, they are only found in the skull. And every single bone of the skull, except one, forms sutures. What's the one bone of the skull that doesn't form a suture? What's the one
Hyoid.
bone of the skull that does not form a suture?
Mandible.
There you go, exactly. The mandible, because it moves, right? Now, every single suture that is formed in the skull has a name. Are you responsible for all of them?
No.
How many sutures do you need to know?
4.
These are the 4 most major ones, and conveniently enough for us, they all involve the parietal bone. Let's start easy. Here you got a parietal bone over here. And you have a parietal bone over here. If we're looking down from the top from a superior view, there happens to be a suture that connects the two parietal bones together. And what would that suture that combines the two parietal bones together be?
Lambdoidal.
Basically, it goes right down the center of the skull, kind of if we did a section of the bone along this
sagittal
suture, it would be
suture?
a sagittal section. And so this is the sagittal suture. Excellent. Anteriorly, there is a suture that connects the two parietal bones to the frontal bone. And what do we call that one where if we cut it, it would be like a frontal section? Or what was the other name for a frontal section? There you go. Sam.
Coronal.
This would be a coronal section. posteriorly, the two parietal bones connect to the occipital bone. And I think the group that presented the occipital bone mentioned this earlier. Notice it forms it in kind of an upside down V shape. And what is the Greek letter that looks like an upside down V?
Lambda. So
Lambda. So this is indeed the lambdoid or lambdoidal. Lambdoid or lambdoidal suture. Oh, that should say suture up there as well. Hold on. Excellent. The last one is a teeny bit tricky because it's not as obvious when I'm drawing it here from the top-down view, but there is a suture between the parietal bone and the temporal bones. And what is that suture between the parietal bone and the temporal bone called?
Squamosal suture.
It's called the squamosal suture. Now, it is called a squamosal suture because, as it turns out, when the temporal bone comes down, it has a very long, flat, oblique surface to it. And when the when this parietal bone comes down, big flat surface, temporal bone has a big flat surface. So they have these two big flat, and of course, squamosal means flat. However, if we look at the picture of this from the side, oh, and here we go. See, there's that nice lambdoidal suture. Here's our sagittal and our coronal. Here is the squamosal. And again, when you look at it here, it's hard to see the two flat surfaces. But what I like about it that makes it easy for me to remember is notice there's all this hunk and junk going on down here with the temporal bone. And the part up here that forms the joint with the parietal bone, this part of the temporal bone is all flat. And so the flat part of the temporal bone forms the flat suture or squamosal suture. So there you go. That is the rest of the skull features and the bones that form them. Any questions on that? All right, with that, now, thankfully, we are done with the skull. So what we are able to do now is to go on to the rest of the axial skeleton. And so let's go ahead and do that after the break. I want to make sure we have enough time. I'll put you guys into your groups again, but let's not spend as much time. Let's just go ahead and take a 20-minute break. So we will come back at 10:55. At 10:55, we will start the group. So here, let me write this in the chat so we all remember. Restart at 10:55. I will go ahead and break you guys into your groups now. Take your break, and then use this time to get organized. And we will finish off the rest of our axial group presentations now. And then from there, we will move on to assigning our appendicular bones that you guys are responsible for. All right, so let's go ahead. Any questions before we take this last break? All right, excellent. I'll meet you back here at 10:55. Go ahead and take your break if you want, and I need a minute or two, and I'll get you guys broken into your groups. So I'll do that right now and see you guys at 10:55.